What are the basic steps to transfer bacteria using aseptic procedures?
First, plug the test tube or flask with cotton or plastic cap. Sterilize the inoculating loop or needle, tool used to transfer the bacteria. By heating the wire in the flame of a Bunsen burner or alcohol lamp until red hot. Remove the cotton plugs from the test tubes or flasks. And briefly flame the mouths of the test tubes or flasks. Pick up the organisms to be transferred by dipping the inoculating loop or needle into the liquid culture or touching the growth lightly. Transfer the culture to a new test tube by dipping the loop gently into the broth or drawing it across the agar. Pass the mouths of the tubes through the flame again and then reinsert the cotton plugs to prevent the entrance of other microbes. Heat the inoculating loop or needle again to destroy any remaining organisms. The procedure to transfer a sample to a petri dish is similar, except the culture is streaked along the agar in a pattern to isolate the bacteria on the petri dish.
How is a fairy ring formed?
Long ago it was believed that the circles of mushrooms that sometimes form in meadows marked the locations where fairies gathered at night to dance. Fairy rings, or fungus rings, are frequently found in grassy areas. There are three types of rings: those that do not affect their surrounding vegetation, those that cause increased vegetational growth, and those that damage their surrounding environment. The rings are started from a mycelium, the underground, food-absorbing part of a fungus. The fungus growths are circular because a round inner band of decaying mycelium forms underground. This band uses up the resources present in the soil that is directly above it. When the fungus forms caps that present above ground, the mushrooms grow around the mycelium, creating a ring effect. Each succeeding generation grows further from the center.
What is a bug, biologically speaking?
The biological meaning of the word bug is significantly more restrictive than in common usage. People often refer to all insects as bugs, even using the word to include such organisms as bacteria and viruses, as well as glitches in computer programs. In the strictest biological sense, a bug is a member of the order Hemiptera, also called true bugs. Members of Hemiptera include bedbugs, squash bugs, clinch bugs, stink bugs, and water striders.
What club moss is called the resurrection plant?
Selaginella lepidophylla, found in the deserts of the southwestern United States and 170 Mexico. is called the resurrection plant because of its ability to defy severe drought conditions. During periods of drought, this plant forms a tight, dried-up ball. When rain comes, its branches expand, become green, and carry out photosynthesis.
What are the differences between an open and a closed circulatory system?
In an open circulatory system, found in many invertebrates, e.g., spiders, crayfish, and grasshoppers, the blood is not always contained within the blood vessels. Periodically, the blood leaves the blood vessels to bathe the tissues with blood and then returns to the heart. There is no interstitial body fluid separate from the blood. A closed circulatory system, also called a cardiovascular system, is found in all vertebrate animals and many invertebrates. In a closed system, the blood never leaves the blood vessels.
How many human RNA genes are there?
There are about 250 genes that code for short RNA sequences instead of protein. These RNA strands appear to regulate the activity of other genes, particularly those involved in embryonic development.
How do air-breathing mammals such as whales and seals dive underwater for extended periods of time?
Seals and whales are able to dive underwater for extended periods of time because they are able to store oxygen. While humans store 36% of their oxygen in their lungs and 51% in the blood, seals store only approximately 5% of their oxygen in their lungs and 70% in their blood. They also store more oxygen in the muscle tissue, 25%, compared with only 13% in human muscle tissue. While underwater, these mammals' heart rates and oxygen consumption rates decrease, allowing some species to remain underwater for up to 20 minutes at a time.
What is the significance of bee dancing? How is it that within a short time of a bee finding a food source, a great number of fellow bees will converge to gather pollen and nectar?
Communication about the food location occurs through the bee dance. This symbolic language of bees, also known as the waggle dance, is the most complex of all non-human communication systems. By placing sources of food at varying distances and angles from the hives, Carl von Frisch found that forager bees perform either a round dance for food at short distances, 66 to 656 feet or 20 to 200 m, or a waggle dance for food at longer distances. While dancing, the forager also emits sound bursts. The specifics of the dance. Example area: the dance occupies on the comb, duration of each cycle, and length of sound bursts, indicate the distance and direction to the food source.
What are the three main types of skeletal systems?
The three main types of skeletal systems are hydrostatic skeleton, exoskeleton, and endoskeleton. A hydrostatic skeleton consists of fluid under pressure. This type of skeletal system is most common in soft, flexible animals such as hydras, planarians, and earthworms and other segmented worms. Hydras and planarians have a fluid-filled gastrovascular cavity. The body cavity, or chylome, of an earthworm is also fluid-filled. Many aquatic and certain terrestrial animals have an exoskeleton. The exoskeleton is rigid and hard. Mollusks have an exoskeleton made of calcium carbonate. It grows with the animal during its entire lifetime. Another type of exoskeleton common among insects and arthropods is made from chitin. Chitin is a strong flexible nitrogenous polysaccharide. While it provides excellent protection and allows for a large variety of movements, it does not grow with the animal. When an animal outgrows its skeleton, it must shed its skeleton and replace it with a larger one in a process known as molding. An endoskeleton consists of bone and cartilage and grows with the animal throughout its life. It stores calcium salts and blood cells and consists of hard or leathery supporting elements situated among the soft tissues of an animal. Although most common among vertebrates, certain invertebrates such as sponges, sea stars, sea urchins, and other echinoderms have an endoskeleton of hard plates beneath their skin. This type of skeletal system allows for a wider range of movement than do the other two.
What are some applications of gas chromatography?
Gas-liquid chromatography is the most widely used chromatographic technique for environmental analyses. Analysis of organic compounds is possible for a variety of matrices such as water, soil, soil gas, and ambient air. It is often used to test hazardous waste sites for determining personal protective equipment. PPE, level and emergency response testing.
What is the cytosol?
The cytosol is the semi-fluid substance of the cell in which the organelles are suspended.
What is the difference between poison ivy, oak, and sumac?
These North American woody plants grow in almost any habitat and are quite similar in appearance. Each variety of plant has 3 leaf compounds that alternate, berry-like fruits, and rusty brown stems. Poison ivy, Rus radicans, grows like a vine rather than a shrub and can grow very high. Covering tall, stationary items such as trees. The fruit of our radicans is gray in color and is without hair, and the leaves of the plant are slightly lobed. Rustoxicodendron, commonly known as poison oak, usually grows as a shrub, but it can also climb. Its leaflets are lobed and resemble the leaves of oak trees, and its fruit is hairy. Poison sumac, Rus vernix, grows only in acidic, wet swamps of North America. This shrub can grow as high as 12 feet 3.6 meters. The fruit it produces hangs in a cluster and ranges from gray to brown in color. Poison sumac has dark green leaves that are sharply pointed, compound, and alternating. It also has inconspicuous flowers that are yellowish green. All parts of poison ivy, poison oak, and poison sumac can cause serious dermatitis.
What is the struggle for existence?
As noted by Thomas Malthus, 1766 to 1834. More individuals are born than can possibly survive due to limited resources. Therefore, there is a struggle for existence. We can think of this as the competition between individuals, of the same species or between species, for food or hiding places or other necessary resources. The origin of this phrase can be traced back to the Greek philosophers but is perhaps best described by Alfred Lord Tennyson's, 1809–1892, phrase, "Nature red in tooth and claw."
What does it mean when a lake is brown or blue?
When a lake is brown, it usually indicates that eutrophication is occurring. This process refers to premature aging of a lake, when nutrients are added to the water. Usually due to runoff, which may be either agricultural or industrial in origin. Due to this rich supply of nutrients, blue-green algae begin to take over the green algae in the lake. And food webs within the lake are disturbed, leading to an eventual loss of fish. When a lake is blue, this usually means that the lake has been damaged by acid precipitation. The gradual drop in pH caused by exposure to acid rain causes disruption of the food webs, eventually killing most organisms. The end result is clear water, which reflects the low productivity of the lake.
When was DNA first discovered?
DNA was first discovered in 1869 by Johann Friedrich Miescher, 1844–1895. He isolated and described what he called nuclein, which eventually became known as deoxyribonucleic acid, DNA, the genetic information of the cell.
Who was the first primate taught to use sign language?
although it was long known that primates use a number of methods of communication in the wild. Early attempts, from 1900 to the 1930s, to teach primates simple words were failures. A 1925 scientific article suggested sign language as an alternative to verbal language in communicating with primates. In the 1960s, researchers tried to teach chimps and gorillas a modified form of sign language. It began with Washoe the chimpanzee, followed by the gorillas Michael, now dead, and Coco. Washoe learned a little over 100 signs. But Coco has a working vocabulary of over 1,000 signs and understands about 2,000 words of spoken English.
How do plants obtain nitrogen?
The primary way that plants obtain nitrogen compounds is via the nitrogen cycle, which is a series of reactions involving several different types of bacteria, including nitrogen-fixing bacteria and denitrifying bacteria. During nitrogen fixation, symbiotic bacteria, which live in association with the roots of legumes, are able through a series of enzymatic reactions to make nitrogen available for plants. Nitrogen is crucial to all organisms because it is an integral element of proteins and nucleic acids. Although Earth's atmosphere is 79% nitrogen, molecular nitrogen is very stable and does not easily combine with other elements. Plants must use nitrogen in its fixed form, such as ammonia, urea, or the nitrate ion.
Who first won the Nobel Prize for work on animal behavior?
Although there is no specific Nobel Prize for the study of animal behavior, the first prize to be awarded for the scientific study of animal behavior was awarded jointly in 1973 in the category Physiology or Medicine to Konrad Lorenz. 1903 to 1989, Carl von Frisch, 1886 to 1982, and Nico Tinbergen, 1907 to 1988. Each ethologist had a main research area for which he was well known, Lorenz for imprinting behavior of birds, von Frisch for the dance of the honeybees, and Tinbergen for aggressive behavior of stickleback fish.
How do cancer cells feed themselves?
In the 1960s, Dr. Judah Folkman, 1933, realized that malignant tumors could not grow without nourishment, which is delivered by the blood. Rapidly growing tumors actually cause the formation of new blood vessels in a process known as angiogenesis. Folkman's hypothesis was that by identifying the substances used to cause angiogenesis, drugs could be formulated to prevent new vessel formation, thus starving the tumors. This work has led to the identification of at least two substances that inhibit angiogenesis, endostatin and angiostatin. These drugs hold promise as new therapies to combat aggressive tumors.
Can cells ever change jobs?
The more specialized of a function a cell performs, the less likely it is for the cell to change jobs within an organism. However, there are some cells that have unspecialized functions and are able to adapt to the changing needs of the body. In mammals, a good example of cells with adapting functions would be bone marrow cells, which are responsible for producing different types of cells in the blood. Bone marrow cells produce red blood cells and 5 types of white blood cells. Slime molds of the kingdom Protista have cells that are capable of drastically changing their function. The cellular adaptations that can occur in slime molds allow them to change from single-celled amoebas to multicellular, reproductive spore producers.
What does this mean?
This means that structurally, the two strands of DNA, the two sides of the ladder, run in opposite directions. This property of antiparallelity allows for formation of the hydrogen bonds between the nitrogenous bases. These bonds are critical to the integrity of the molecule.
What are some of the most common enzyme deficiencies?
Lactose intolerance, a condition that results from the inability to digest the sugar present in milk. Lactose is one of the most common enzyme deficiencies. While nearly everyone is born with the ability to produce lactase, the enzyme responsible for lactose breakdown, many people lose this ability with age. Lactase production in adults varies by ethnic group. Glucose 6-phosphate dehydrogenase deficiency is a more serious enzyme deficiency that is linked to the bursting of red blood cells, hemolysis. This deficiency is found in more than 200 million people, mainly Mediterranean, West African, Middle Eastern, and Southeast Asian populations.
Why are sea urchins used to study embryonic development?
Sea urchins are a useful model system for studying many problems in early animal development. Historically, sea urchins were a key system in elucidating a variety of classic developmental problems, including the mechanisms of fertilization, egg activation, cleavage, gastrulation, and the regulation of differentiation in the early embryo. In addition, early studies of the molecular basis of early embryology were carried out in this system. Gametes can be obtained easily, sterility is not required, and the eggs and early embryos of many species are beautifully transparent. The early development of sea urchin embryos is also highly synchronous, that is, when a batch of eggs is fertilized, all of the resulting embryos typically develop on the same schedule, making possible biochemical and molecular studies of early embryos.
When were chromosomes first observed?
Chromosomes were observed as early as 1872, when Edmund Rousseau, 1841 to 1897, described seeing items that resembled small rods during cell division. He named the rod stab chen. Edouard van Beneden, 1846 to 1910, used the term bâtonnet in 1875-40 to describe nuclear duplication. The following year, 1876, Edouard Balbiani, 1825 to 1899, described that at the time of cell division the nucleus dissolved into a collection of bâtonnet centroids, narrow little rods. Walther Fleming, 1843 to 1905, discovered that the chromosomal threads or phaeton split longitudinally during mitosis.
Can DNA be extracted from a mummy?
Yes, DNA can be, and has been, extracted from a mummy. However, the problem with extraction of ancient DNA lies within contamination from modern DNA. In order to minimize contamination, researchers usually try to get DNA from inside teeth or bone. Ancient DNA is being used to study the genealogy of the pharaohs of Egypt.
How often is the epithelium replaced?
Epithelial cells are constantly being replaced and regenerated during an animal's lifetime. The epidermis, outer layer of the skin, is renewed every 2 weeks, while the epithelial lining of the stomach is replaced every 2 to 3 days. The liver, a gland consisting of epithelial tissue, easily regenerates after portions are removed surgically.
Is it possible to observe evolutionary change in other species?
The change in allele frequencies within populations, which is driven by natural selection, is occurring around us constantly. However, whether we can actually observe those changes is a different matter. For example, elephants may be adapting to changing conditions in their environment. But since they tend to be extremely long-lived, it is unlikely that we will be able to follow enough generations to observe a trend. On the other hand, it is possible to notice such changes in populations of individuals with very short lifespans, particularly when they are experimentally manipulated. Bacteria and guppies are just 2 examples of species that have been observed evolving in response to changing environmental conditions.
What are the best temperatures for seed germination?
The best temperatures for seed germination in most plants are 77 degrees Fahrenheit to 86 degrees Fahrenheit, 25 degrees Celsius to 30 degrees Celsius. Some varieties of seeds are able to germinate at temperatures ranging from 41 degrees Fahrenheit to 86 degrees Fahrenheit, 5 degrees Celsius to 30 degrees Celsius. However, some seeds, such as those of the Rocky Mountain lodgepole pine tree, Pinus contorta, require extreme heat for germination. The cones of the Rocky Mountain lodgepole pine are covered with the resin that binds the scales of the cone. The high temperatures, 113 to 120 degrees Fahrenheit, or 45 to 50 degrees Celsius, associated with 202 moderate to severe forest fires are required to melt the resin and release the seeds.
Why are insects often found in amber?
People have long been infatuated with amber, the fossilized form of ancient tree resin. A semi-precious stone used for jewelry and mosaics. Amber from the Dominican Republic contains an average of 1 insect in every 100 pieces. Some pieces of amber contain thousands of insects, both whole insects and insect fragments. These insects were probably crawling or lodged on the outside of a tree about 30 million years ago and became trapped by a glob of sticky tree resin, which continued to ooze around the animal matter and eventually fossilized. Scientists are able to study these insects, many of which are extinct but may turn out to be missing links to modern-day species.
Which insect has the best sense of smell?
Giant male silk moths, Bombyx mori, may have the best sense of smell in the world. Their antennae are covered with about 65,000 tiny bristles. Most of the bristles are chemoreceptors. The moths can smell a female's perfume nearly 7 miles 11 kilometers away.
What are the main components of the nucleus?
The boundary around the nucleus consists of 2 membranes, an inner one and an outer one, that form the nuclear envelope. Nuclear pores are small openings in the nuclear envelope that permit molecules to move between the nucleus and the cytoplasm. The nucleolus is a prominent structure within the nucleus. The nucleoplasm is the interior space of the nucleus. In addition, the DNA-bearing chromosomes of the cell are found in the nucleus. The nucleus is the repository for the cell's genetic information, and the control center for the expression of that information.
What is the most destructive insect in the world?
The most destructive insect is the desert locust, Schistosera gregaria. The locust of the Bible, whose habitat ranges from the dry and semi-arid regions of Africa and the Middle East through Pakistan and northern India. This shorthorn grasshopper can eat its own weight in food a day. And during long migratory flights a large swarm can consume 20,000 tons, 18,144,000 kilograms, of grain and vegetation a day.
What are some methods of asexual reproduction?
Budding, fission, and fragmentation are methods of asexual reproduction. In budding, a new individual begins as an outgrowth, or bud, of the parent. Eventually, the bud detaches from the parent and develops into a new individual. Budding is common among sponges and coelenterates such as hydras and anemones. Fission is the division of one individual into two or more individuals of almost equal size. Each new individual develops into a mature adult. Some corals reproduce by dividing longitudinally into two smaller but complete individuals. Fragmentation is the breaking of the parent into several pieces. It is accompanied by regeneration when each piece develops into a mature individual. Sea stars are well known for reproducing by fragmentation and regeneration.
Who started Earth Day?
The first Earth Day, April 22, 1970, was coordinated by Dennis Hayes at the request of Gaylord Nelson, 1916, U.S. Senator from Wisconsin. Nelson is sometimes called the father of Earth Day. His main objective was to organize a nationwide public demonstration so large it would get the attention of politicians and force the environmental issue into the political dialogue of the nation. Important official actions that began soon after the celebration of the first Earth Day were the establishment of the Environmental Protection Agency, EPA, the creation of the President's Council on Environmental Quality, and the passage of the Clean Air Act, establishing national air quality standards. In 1995, Gaylord Nelson received the Presidential Medal of Freedom for his contributions to the environmental protection movement. Earth Day continues to be celebrated each spring.
How are tree rings used to date historical events?
The study of tree rings is known as dendrochronology. Every year, trees produce an annular ring composed of one wide, light ring and one narrow, dark ring. During spring and early summer, tree stem cells grow rapidly and larger, thus producing the wide, light ring. In winter, growth is greatly reduced and cells are much smaller, producing the narrow, dark ring. In the coldest part of winter or the dry heat of summer, no cells are produced. Comparing pieces of dead trees of unknown age with the rings of living trees allows scientists to establish the date when the fragment was part of a living tree. This technique has been used to date the ancient pueblos throughout the southwestern United States. A subfield of dendrochronology is dendroclimatology. Scientists study the tree rings of very old trees to determine climatic conditions of the past, the effects of droughts, pollution, insect infestations, fires, volcanoes, and earthquakes are all visible in tree rings.
What are telomeres and where are they located?
Telomeres are protective structures composed of DNA that contain multiple repetitions of one short nucleotide sequence instead of genes. They are found at each end of a eukaryotic chromosome.
What are diatoms?
Diatoms are microscopic algae of the phylum Chrysophyta in the kingdom Protista. Almost all diatoms are single-celled algae and dwell in both fresh and salt water. They are abundant in the cold waters of the northern Pacific Ocean and the Antarctic. Diatoms are yellow or brown in color and are an important food source for marine plankton and many small animals. Diatoms have hard cell walls. These shells are made from silica that has been extracted from the water. It is unclear how the extraction of silica from water is accomplished. When they die, their glassy shells called frustules sink to the bottom of the sea and harden into rock called diatomite. One of the most famous and accessible diatomites is the Monterey Formation along the coast of central and southern California.
What are demyelinating diseases?
Demyelinating diseases involve damage to the myelin sheath of neurons in either the peripheral or central nervous system. Multiple sclerosis, MS, is a chronic, potentially debilitating disease that affects the myelin sheath of the central nervous system. The illness is probably an autoimmune disease. In MS, the body directs antibodies and white blood cells against proteins in the myelin sheath, surrounding nerves in the brain and spinal cord. This causes inflammation and injury to the myelin sheath. Demyelination is the term used for a loss of myelin, a substance in the white matter that insulates nerve endings. Myelin helps the nerves receive and interpret messages from the brain at maximum speed. When nerve endings lose this substance, they cannot function properly, leading to patches of scarring, or sclerosis. The result may be multiple areas of sclerosis. The damage slows or blocks muscle coordination, visual sensation, and other functions that rely on nerve signals. In the autoimmune disorder known as Guillain-Barré syndrome, the body's immune system attacks part of the peripheral nervous system. The immune system starts to destroy the myelin sheath that surrounds the axons of many peripheral nerves, or even the axons themselves. The myelin sheath surrounding the axons speeds up the transmission of nerve signals and allows the transmission of signals over long distances. In diseases such as Guillain-Barré, in which the peripheral nerves' myelin sheaths are injured or degraded, the nerves cannot transmit signals efficiently. Consequently, muscles begin to lose their ability to respond to the brain's commands. Commands that must be carried through the nerve network. The brain also receives fewer sensory signals from the rest of the body, resulting in an inability to feel textures, heat, pain, and other sensations. Alternately, the brain may receive inappropriate signals that result in tingling, crawling skin, or painful sensations. Because the signals to and from the arms and legs must travel the longest distances, these extremities are most vulnerable to interruption. The first symptoms of this disorder include varying degrees of weakness or tingling sensations in the legs. In many instances, the weakness and abnormal sensations spread to the arms and upper body. In severe cases, the patient may be almost totally paralyzed since the muscles cannot be used at all. In these cases, the disorder is life-threatening, potentially interfering with breathing and, at times, with blood pressure or heart rate, and is considered a medical emergency. Such a patient is often put on a respirator to assist with breathing and is watched closely for problems such as an abnormal heartbeat, infections, blood clots, and high or low blood pressure. Most patients, however, recover from even the most severe cases of Guillain-Barré syndrome, although some continue to have a certain degree of weakness.
How do fleas jump so far?
The jumping power of fleas comes both from strong leg muscles and from pads of a rubber-like protein called resilin. The resilin is located above the flea's hind legs. To jump, the flea crouches, squeezing the resilin, and then it relaxes certain muscles. Stored energy from the resilin works like a spring, launching the flea. A flea can jump well both vertically and horizontally. Some species can jump 150 times their own length. To match that record, a human would have to spring over the length of 2.25 football fields or the height of a 100-story building in a single bound. The common flea, Pulex irritans, has been known to jump 13.33 centimeters in length and 18.4 centimeters in height.
How does one distinguish between thorns, spines, and prickles?
Thorns are modified branches or stems arising from the axils of leaves. One purpose of thorns is to protect the plant from grazing animals. Hawthorn trees are an example of a plant with true thorns. Spines are modified leaves such as those found on cactus plants. Prickles are sharp outgrowths from the epidermis of various plant structures including leaves and stems.
What is biofarming?
Biofarming is a relatively new technology in which transgenic plants are used to grow pharmaceuticals. In this technique, scientists bioengineer medically important proteins into a corn plant, thus spurring the corn plant to produce large amounts of a particular medicinal protein. This is less expensive than using a microbial fermentation chamber.
Can animals behave altruistically?
Altruism is the performance of a behavior that will benefit the recipient at a cost to the donor, such as risking one's life to save another. There are numerous examples of animals exhibiting altruistic behavior. For instance, adult crows may act as nannies for other crows. Instead of increasing their fitness by producing their own offspring. Another example is a ground squirrel that will warn others of the presence of a predator, even though making such a call may draw the attention of the predator to itself. In studying social insects, Edward O. Wilson, 1929, found that in many species of social insects, workers forego reproduction entirely, they are sterile, in order to help raise their sisters. There are two possible explanations for this behavior. The donor is performing the act either in hope that the recipient will someday return the favor, reciprocal altruism, or because the recipient is a family member. In the game of evolution, winners are those who leave the greatest number of copies of their genes in the subsequent generations. So this kin selection form of altruism may not be so altruistic after all.
What is El Niño?
Along the west coast of South America, near the end of each calendar year, a warm current of nutrient-poor tropical water moves southward, replacing the cold, nutrient-rich surface water. Because this condition frequently occurs around Christmas, local residents call it El Niño. Spanish for child, referring to the Christ child. In most years the warming lasts for only a few weeks. However, when El Niño conditions last for many months, the economic results can be catastrophic. It is this extended episode of extremely warm water that scientists now refer to as El Niño. During a severe El Niño, large numbers of fishes and marine plants may die. Decomposition of the dead material depletes the water's oxygen supply. which leads to the bacterial production of huge amounts of smelly hydrogen sulfide. A greatly reduced fish, especially anchovy, harvest affects the world's fish meal supply, leading to higher prices for poultry and other animals that normally are fed fish meal. Anchovies and sardines are also major food sources for marine mammals such as sea lions and seals. When the food source is in short supply, these animals travel further from their homes in search of food. Not only do many sea lions and seals starve, but also a large proportion of the infant animals die. During the winter of 1997 to 1998, El Niño effects resulted in the second warmest and seventh wettest winter since 1895. Severe weather events included flooding in the U.S. Southeast, ice storms in the Northeast, flooding in California, and tornadoes in Florida. The 1997 to 1998 event indirectly caused 2,100 deaths and $33 billion in damage globally.
Where are ribosomes found in a cell?
Ribosomes are found in the cytoplasm of both prokaryotic and eukaryotic cells, as well as in the matrix of mitochondria and the stroma of chloroplasts. In eukaryotic cytoplasm, ribosomes are found in the cystole and are bound to the endoplasmic reticulum as well as the outer membrane of the nuclear envelope.
Can cells be seen without a microscope?
Yes, but not many. Most cells are far smaller than the period at the end of the sentence. Bird and frog egg cells may be somewhat larger and can in fact be observed by the unaided eye.
What is meant by a dominant animal?
When two animals attempt to acquire the same resource simultaneously, e.g., a food item, and one consistently wins the prize, that animal is considered dominant. Evolutionary theory suggests that dominance may be a way for animals to avoid actual fighting, since aggressive interaction may be physically costly even for the winner.
What is the Sanger technique?
The Sanger technique was developed by Fred Sanger, 1918, in the 1970s. It is also referred to as shotgun sequencing and is a common method used for DNA sequencing. The basics of this method involve using dideoxynucleotides instead of the deoxynucleotides normally found in DNA. Dideoxynucleotides are nucleotides missing a hydroxyl group at the site where one nucleotide usually joins to another nucleotide. If the hydroxyl group is missing, chain elongation stops. The process can be done manually or by automation. The cost of sequencer is approximately $100,000. In the automated process, a short DNA primer is synthesized that is complementary to the template to be analyzed. Electrophoresis is used to separate the bands produced. A different fluorescent label is used on each of the dideoxynucleotides. Adenine, guanine, thymine, cytosine, and these are added to the template. Once hybridization occurs, the chain elongation stops and large numbers of fluorescent bands are produced and read using a laser. The laser then transmits the sequence into nitrogen bases.
What is the relationship between fungi and ants?
The leaf-cutting ants, found in Central and South America as well as in the southern United States, have a mutually beneficial, or symbiotic, relationship with certain fungi of the genus Septobasidium. The ants are not able to digest the cellulose found in leaves. The fungus breaks down the cellulose, a food source for the fungus, and converts it into carbohydrates and proteins, which the ants can digest. The ants then eat the fungus. The ants provide the fungus with a guaranteed food supply and eliminate competing fungi. The ants and fungus are not known to occur independently from each other.
What are the specialized cells in plants?
All plant cells have several common features, such as chloroplasts, a cell wall, and a large vacuole. In addition, a number of specialized cells are found only in vascular plants. They include parenchyma cells, parenchyma, from the Greek para, meaning beside, and enchine, meaning to pour in. Cells are the most common cells found in leaves, stems, and roots. They are often spherical in shape with only primary cell walls. Parenchyma cells play a role in food storage, photosynthesis, and aerobic respiration. They are living cells at maturity. Most nutrients in plants, such as corn and potatoes, are contained in starch-laden parenchyma cells. These cells comprise the photosynthetic tissue of a leaf, the flesh of fruit, and the storage tissue of roots and seeds. Collenchyma cells, collenchyma, from the Greek term kolla, meaning glue. Cells have thickened primary cell walls and lack secondary cell walls. They form strands or continuous cylinders just below the surfaces of stems or leaf stalks. The most common function of collenchyma cells is to provide support for parts of the plant that are still growing, such as the stem. Similar to parenchyma cells, collenchyma cells are living cells at maturity. Sclerenchyma cells, sclerenchyma, from the Greek term skleros, meaning hard, cells have tough, rigid, thick secondary cell walls. These secondary cell walls are hardened with lignin, which is the main chemical component of wood. It makes the cell walls more rigid. Sclerenchyma cells provide rigid support for the plant. There are two types of sclerenchyma cells: fiber and sclereid. Fiber cells are long, slender cells that usually form strands or bundles. Sclereid cells, sometimes called stone cells, occur singly or in groups and have various forms. They have a thick, very hard secondary cell wall. Most sclerenchyma cells are dead cells at maturity. Xylem. Xylem, from the Greek term xylos, meaning wood, is the main water-conducting tissue of plants and consists of dead, hollow, tubular cells arranged end to end. The water transported in xylem replaces that lost via evaporation through stomata. The two types of water-conducting cells are tracheids and vessel elements. Water flows from the roots of a plant up through the shoot via pits in the secondary walls of the tracheids. Vessel elements have perforations in their end walls to allow the water to flow between cells. Phloem. The two kinds of cells in the food-conducting tissue of plants, the phloem, from the Greek term phloios, meaning bark, are sieve cells and sieve tube members. Sieve cells are found in seedless vascular plants and gymnosperms, while sieve tube members are found in angiosperms. Both types of cells are elongated, slender, tube-like cells arranged end to end with clusters of pores at each cell junction. Sugars, especially sucrose, other compounds, and some mineral ions move between adjacent food-conducting cells. Sieve tube members have thin primary cell walls but lack secondary cell walls. They are living cells at maturity. Epidermis. Several types of specialized cells occur in the epidermis, including guard cells, trichomes, and root hairs. Flattened epidermal cells, one layer thick and coated by a thick layer of cuticle, cover all parts of the primary plant body.
How does the nucleus of an atom differ from the nucleus of a cell?
The English word nucleus, meaning inside the shell, is derived from the Latin word for nut. The nucleus of an atom is an enclosed space containing protons and neutrons. The nucleus of a cell is a membrane-enclosed organelle that contains the genetic material of a cell.
Do fungi exhibit an alternation of generations similar to plants?
Organisms with life cycles that exhibit an alternation of generations have both a haploid phase and a diploid phase. During the haploid phase, reproduction is conducted through gametes, which fuse together to form a zygote. During the diploid phase, reproduction is conducted by spores. Spores develop individually and divide through mitosis, producing the gametophytes of the next generation. Because fungi have both a haploid and diploid phase, they exhibit an alternation of generations.
What is the periderm?
Periderm replaces the epidermis as the protective covering. In stems and roots of plants with secondary growth. The periderm consists of three structures: 1. The cork or phellum. 2. The cork cambium or phellogen, and 3. The phelloderm. The cork or phellum is non-living and is the protective tissue formed to the outside by the cork cambium. The cork cambium or phellogen is the meristem that produces the periderm. The phelloderm is a living parenchyma tissue formed to the inside of the meristem.
How are organelles partitioned during mitosis?
Following the telophase of mitosis, cytokinesis, physical separation of the daughter cells, occurs. Although the exact mechanisms are not clear, it appears that larger organelles, such as the endoplasmic reticulum and Golgi complex, undergo fragmentation into small vesicles during mitosis and later reassemble in daughter cells.
Is alcoholism an inherited disease?
If all of the adults in the U.S. were to be tested, an estimated 10% of the adult population would be classified as alcoholic. Of that number, the ratio of males to females is approximately 4:1. This disproportion could be attributed to both environment and genetics. Research involving male relatives, both biological and adoptive, points to a multifactorial source for alcoholism. In other words, while sons and brothers of male alcoholics are likely to be alcoholic as well, 25% and 50%, respectively. Genetic inheritance is not solely responsible for all of the cases of alcoholism. Alcoholism has been linked to depression, as more is learned about the genetic basis of mood disorders. The genetic basis of alcoholism may be unraveled.
How do fungi cause trees to become hollow?
Trees with hollow interiors are a familiar sight in parks, forests, and throughout urban areas. Fungal decay is often the cause of the hollowing of the trees. Wood-decaying fungi can enter a tree following a wound to the tree. The tree will produce a band of cells that tries to resist the formation of decay directly around the wound. The fungus that has entered the tree through the wood will continue to decay the inner lichens. Like the yellow lichen shown here, are often used as natural dyes and in the perfume and cosmetic industries. Wood, while the wood protected by the band of cells will continue to be sound. Hollow trees are also formed by fungi that solely attack the hardwood of a tree. This is most commonly observed in older coniferous trees of the Pacific coast of North America, reaching from California to Alaska. Heart rot fungi can enter a tree either through a wound or through small branch stubs. It decays the inner wood, resulting in a weak tree that is susceptible to damage by strong winds.
What is Gattaca?
The 1997 film Gattaca was one of the first movies to examine the idea that one's station in life is dependent on the perfection of one's genes. The movie explores genetic discrimination and previews the bioethics of using genetic information as a means of identification.
How large is the arthropod population?
Zoologists estimate that the arthropod population of the world, including crustaceans, spiders, and insects, numbers about a billion million, 1018, individuals. More than 1 million arthropod species have been described, with insects making up the vast majority of them. In fact, 2 out of every 3 organisms known on Earth are arthropods. And the phylum is represented in nearly all habitats of the biosphere. About 90% of all arthropods are insects, and about half of the named species of insects are beetles.
What were early ideas on evolution?
While some Greek philosophers had theories about the gradual evolution of life, Plato, 427 to 347 b. CE, and Aristotle, 384 to 322 BCE, were not among them. In the 1700s, natural theology, the explanation of life as the manifestation of the Creator's plan, held sway in Europe. This idea was the motive force behind the work of Carl Linnaeus, 1707 to 1778. Who was the first to classify all known living things by kingdom. Also popular prior to the work of Charles Darwin, 1809 to 1882, were the theories of special creation. Creationism, blending inheritance. That offspring were always the mixture of the traits of their 2 parents, and acquired characteristics.
How do hardwoods differ from softwoods?
Hardwood and softwood are terms used commercially to distinguish woods. Hardwoods are the woods of dicots, regardless of how hard or soft they are, while softwoods are the woods of conifers. Many hardwoods come from the tropics, while almost all softwoods come from the forests of the northern temperate zone. The carotenoids, pigments in the photosynthesizing cells, which are responsible for the fall colors, are present in the leaves during the growing season. However, the colors are eclipsed by the green chlorophyll. Toward the end of summer, when the chlorophyll production ceases, the other colors of the carotenoids, such as yellow, orange, red, or purple, become visible. Listed below are the autumn leaf colors of some common trees.
What is the difference between regular acidophilus milk and sweet acidophilus milk?
Both acidophilus milk and sweet acidophilus milk are inoculated with the bacterium Lactobacillus acidophilus. Many health practitioners believe that this bacterium, a normal member of the human intestinal flora, aids in digestion. Regular acidophilus milk is produced by adding the Bacterium to vats of skim milk as part of the fermentation process. It has a characteristic sour taste. When the bacterium is added to pasteurized milk and packaged without fermentation, sweet acidophilus milk, which lacks the characteristic sour taste, is produced.